Hello everyone. Last lesson, we ended on the topic of ploidy, which is the number of chromosomes set for a cell or a specific species. For example, for us humans, we have 46 chromosomes in one human body cell. So that makes us a diploid or a 2N. Now, before we dive to the different stages of cell division, particularly mitosis, it's important that we know which phase of the cell cycle it is. The cell cycle, again, is a series of events that a cell goes through as it grows and divides. It is composed of the three phases or three main events: the interphase, the mitosis or M phase, and cytokinesis. The interphase is known as the non-dividing phase. This is the process in which a cell may double its entire content in preparation for cell division. Next, we have the M phase, which is the dividing phase. This is where the cell contents are distributed into daughter cells. And lastly, we have cytokinesis or cytoplasmic division. It is the final step in cell division where the cytoplasm of a single eukaryotic cell divides into two daughter cells. Now, there are three phases in interphase. First, we have the G1 phase. Here, the cell grows. So that's basically it. That's what the cell does in the G1 phase. Here, the nucleus and cytoplasm are enlarging towards mature size. So that means the cell increases in volume. So it does so by imbibing water and nutrients into the cell. Here, the chromatin is stretched into a long chromatin fiber. So at the G1 phase, the chromatin is starting to get folded or to get condensed into chromatin fiber as a preparation for it to become a chromosome. Next, we have the S phase. The S phase is a stage or phase in which active synthesis of DNA and associated proteins such as the histones happen. So the DNA is doubled after S phase, or it is replicated, which means that each chromatin fiber is also replicated and doubled. So that's why it's called an S phase, because active synthesis of DNA and histones happen in this phase or in this stage. Lastly, we have the G2 phase or the gap2 phase. So in the G2 phase, active synthesis of RNA and proteins necessary for chromosome synthesis and mitosis happens. The double chromatin fiber is now folded to form a chromosome. So from being a chromatin in G1 phase, it becomes a chromatin fiber, and then this chromatin fiber is replicated and doubled during the S phase, and then at the G2 phase, this chromatin fiber is folded to become a chromosome. So at this stage, each of the chromosomes is composed of a sister chromatids. So now we have this at the G2 phase. Now, what do you think happens in each stage of the cycle? So here is the cell cycle basically. First, we have the interphase, and then the stages or the different stages of mitosis. So the first stage in mitosis is the prophase. Let's write M1. Then the second stage is prometaphase, and then metaphase, and then anaphase, and then telophase. And then telophase, after telophase, we have cytokinesis, and then the cell formed after cytokinesis, or the daughter cells formed after cytokinesis, will again undergo interphase, and the cycle repeats. So let's start with the interface. In the cell cycle, the G1 phase or the first gap phase is where again the cell grows and gets ready to copy its DNA. And unlike other phases, G1 can vary a lot in length depending on the type of cell or its environment. So, sometimes instead of continuing through the cell cycle, a cell in G1 will enter a resting phase or what we call the G0 phase. So this is the resting phase. In G0 phase, the cell stops dividing and just carries out its normal function. Some cells can stay in G0 phase permanently. So for example, the nerve or heart cells. The nerve cells doesn't divide after it grows and mature. It just stays as a nerve cell and does its job as a normal nerve cell. On the other hand, some cells can re-enter G1 again and start dividing again if needed. So for example, our red blood cells. So they can re-enter G1 and start dividing again if they need to do so. The cell doesn't move through the cycle randomly. It is carefully controlled by internal signals such as proteins and different checkpoints that we will discuss later. So these regulations will make sure that the cell is ready before moving on to the next phase, and it helps control how many times a cell divides. And also, it prevents errors that could lead to problems such as cancer. Now, here are the 5 stages or 5 sequential stages of mitosis. So, first, we have the prophase, and then the prometaphase, and then the metaphase, anaphase, telophase. And then lastly, we have the cytoplasmic division or the cytokinesis, which coincides with telophase. So normally when you look at a cell undergoing mitosis on using a microscope, prometaphase is not really seen or viewed during that process. What we can only see is this 4 process: prophase, metaphase, anaphase, telophase, and then sometimes cytokinesis. But remember that prometaphase is still included in the 5 stages of mitosis. Now, ma'am, what happens in prophase? So here's what happens during prophase. So the chromosomes shorten, thicken, and become visible as thick rods. So from being long, like that, the chromosome will condense. Okay, so this is actually wrong. Baliktad sila. So the chromatin will condense into chromosomes. So this is the chromatin, and then it will condense into chromosomes, or it will shorten, thicken, and become visible as thick rods under the microscope. So it will transform to this familiar thing, or this chromosome. So they coil and shorten, or the chromosomes coil and shorten, because it is easier to divide the contents of the cells when they are compressed or when they are small, since as we know, chromatins are very, very, very long. So they are coiled and shortened for efficiency and for easier division of contents. By late prophase, the nucleoli and nuclear membrane disappears, and spindle fibers start to form from the centrosome. And then it will radiate towards the opposite ends of the cell, where they will form spindle poles. Mom, what are spindle fibers? I will write it down. Spindle fibers are actually microtubules that separates the chromosomes. So these spindle fibers or mitotic spindle are the ones to pull the chromosomes apart. Okay. And then this is what prophase looks like. So here, as you can see, the nuclear membrane and the nucleoli of the nucleus is starting to disappear. And then the early mitotic spindle is beginning to form from the centrosome. Next, after prophase, we have the prometaphase. So the prometaphase is a transitional stage within mitosis. It occurs after prophase and before metaphase. That's why it's a transitional stage. And prometaphase is all about making sure that the chromosomes are ready to be moved to the opposite ends of the cell. So, the nuclear envelope or the nuclear membrane, which encloses the nucleus, completely disintegrates, releasing the chromosomes into the cytoplasm. And then proteins will assemble at the centromere region of each chromosome. If you remember, the centromere is this primary constriction here at the middle of our chromosome. That's the centromere. So the proteins will assemble at the centromere and it will form the structures called kinetochores. So we have kinetochore 1 and then kinetochore 2, one for each side of the centromere. Then the microtubules, which are part of the mitotic spindle, will extend from the poles of the cell and attach to the kinetochores. So from the centrosome, the mitotic spindle will attach to the kinetochore, or the kinetochore microtubules will attach to the kinetochore. So the chromosomes are then moved by these microtubules, undergoing a back and forth motion as they are pulled towards the poles. So parang nagsiswing pa siya during prometaphase. And then after prometaphase, we have metaphase. So here, the chromosomes, which are maximally condensed, aligned at the equatorial plane or the metaphase plate. So this broken line here is what we call the metaphase plate. The chromatids in each chromosome separate from each other except at the centromere. The arms of the chromatids may slightly separate, but they remain joined at the centromere, the central point where they are connected. So let me draw what I just described or what I just said. So for example, this is the two poles of our cell, and then we have these chromosomes. So again, during metaphase, the mitotic spindle will attach to the centromere, or to more specifically to the kinetochore attached to the centromere. There. And then they will align it, or the chromosomes, at the metaphase plate. So here is our metaphase plate at the center. So the arms may slightly separate, or like from being this, they will become like that, extended like that. So they may slightly separate, but they are still attached together at the centromere, or at the center, or at the primary constriction. So the cell checks that all chromosomes are aligned properly and correctly attached to the spindle. So this is what we call the metaphase checkpoint, which we will discuss later. This will prevent errors the cell before the cell proceeds to anaphase. So here's what will happen to anaphase. If at metaphase, this is what the cell looked like. Excuse my poor drawing. So at anaphase, this is what will happen. The spindle fibers or the mitotic spindle will pull the sister chromatids or the chromosomes apart. So it's a tragic love story. So they will pull these sister chromatids towards the pole or towards the opposite pole. So at anaphase, the centromeres become functionally double because before anaphase, they function as one. Each member of the doubled chromosomes begins to move toward the opposite poles as I described earlier, and this poleward movement is actually due to the polymerization of the spindle fibers, where the spindle fibers gets shorter and shorter. That's why these sister chromatids are pulled towards the opposite poles of the cells. After anaphase, we have telophase. So this is what again our cell will look like at anaphase. We have the separated sister chromatids located at the different or at the opposite poles of our cell. So at telophase, this is what will happen. The spindle disassembles and chromosomes at each spindle pole decondense, returning to the extended state typical of interphase. So the mitotic spindle will start to get removed or erased. Then the cell will look like an apple. Yan. And then nucleolus reappears. So we will put a circle there at the opposite ends of the cell, and RNA transcription resumes. And according to the earlier description, chromosomes at each spindle pole decondense, or that means from being a chromosome, they will go back to becoming a chromatin fiber or a chromatin. So again, they will become like these long things. So a new nuclear envelope forms, or a new nuclear membrane will form like that at each pole, producing the two daughter nuclei. So at telophase, we now have two daughter nuclei or one daughter nucleus per each cell. So this is cell 1, this is cell 2. At this point, nuclear division is complete since the cell has two nuclei, but we're not quite done yet because we still have cytokinesis or cytoplasmic division, the last stage of the cell cycle. So the cytokinesis or the cytoplasmic division, as stated earlier, produces two daughter cells, each with one of the two daughter nuclei. So the plane of cytoplasmic division, or the location where the cytokinesis will cut the cell in half, is determined by the layer of microtubules that persists at the former spindle midpoint. So usually the metaphase plate is also the plane for cytoplasmic division. There are two types of cytokinesis. One is by farrowing and one is by cell plate formation. So farrowing is observed in animals, protists, and many fungi. So here's what happened when cytokinesis is by farrowing. So the location of the cleavage furrow is again determined by the mitotic spindle, and this ensures that division occurs at the midpoint between the two daughter nuclei. And then a contractile ring will form, or this thing that is what we call the contractile ring. So the contractile ring will gradually contract, which will cause the cell membrane to form a furrow, or what we call the cleavage furrow. This will pinch the cell into two. So this cleavage furrow will deepen until the two daughter cells are completely separated by the constriction of the contractile ring and the fusion of the membranes. Okay, sorry. So this is the contractile ring and this is the cleavage furrow. So again, what happens is that the contractile ring will continue to contract until it forms a cleavage furrow. This furrow will deepen into the two daughter cells, effectively separating the two daughter cells. Next, we have cell plate formation. So cell plate formation actually happens in plant cells. So here, a layer of vesicles containing wall material collects in the plane of the former spindle midpoint. So here is the former spindle midpoint at the middle. So more vesicles are added to this layer until it extends across the cell, and then these vesicles will fuse together, and then it will continue until the two daughter nuclei are separated into two cells. So basically what happens is that a layer of vesicles will form at the spindle midpoint, and then more vesicles will be added to that initial or a starting layer, and then these vesicles will thicken and fuse together. So this fusion will continue until the two daughter nuclei are finally separated by a cell wall. So that's it for cytokinesis. I hope you learned something and that 5 different stages of mitosis was clear to you. So again, if you want a recap, first we have the pro-metaphase. So here we still have our chromatin fiber. They are still condensing to become a thick chromosome. So that's the pro-metaphase. And then we have the metaphase. So at metaphase, our chromosomes will align at the metaphase plate, and mitotic spindle fibers will attach to the kinetochore located at the centromere or at the center of our chromosomes. So after metaphase, we have anaphase. So at anaphase, the sister chromatids of each chromosome will be separated or pulled towards the opposite pole. And then lastly, we have telophase. So at telophase, the nuclei will form again, which will contain the decondensed chromosomes or the chromatin, and then two daughter cells are produced, and then cytokinesis will follow, which will separate the two daughter cells. So that's it. That's the stages of mitosis. Goodbye and thank you. Please watch our next video lecture, which is about the different checkpoints that you can find in the cell cycle. Bye bye.